From Microbe TV, this is TWiEVO, This Week in Evolution, episode number 58, recorded on August 12th, 2020. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels LD.
Hey, Vincent. Great to be with you from LD Home Studio and Nursery.
Combination, multi-use room, right?
Yeah, we added a sort of a new wing here with a little one on the way, or here has arrived. Actually, 2 months old now.
Cool.
Fresh daughter.
You getting any sleep?
You know, not a lot, but enough to make it through. And actually, the nursery isn't functioning yet. Yeah. Well, it's a studio at the moment. It's so warm here in the summer. Salt Lake, and I'm on the upper floor, which in this old house is sort of on the warm side. So currently more recording than baby nurturing at the moment. So, and Vincent, I heard a rumor that I'm not the only sort of TWiV adjacent podcaster with a little one in the mix. Is that true, Stephanie, from your other podcast, Immune?
Yeah. She and her husband had a baby, I don't know, not too long ago.
Yeah.
Maybe
I think I
July.
just saw a tweet of an adorable infant boy, Graham, looking good.
Yeah, it's cool. Well, on the two out of 6 podcasts, people had babies. It's cool.
Pandemic infants here.
Yep.
I'm looking forward to the TWiV picnic once the pandemic's over.
That's right. We'll get together. Well, today we have 2 guests from my campus, Columbia University, from the Center for Infection and Immunity. That's part of the School of Public Health at Columbia University. Simon Anthony, welcome to TWiEVO, Simon.
Thank you very much. Thanks for having me.
Pleasure. And also from the CII, Heather Wells. Welcome, Heather.
Thank you, Vincent. Thank you.
Although Heather is in Ohio, I understand, right?
Yes.
When did you go out there?
I left, I think, the second week of March. So I've been here for 4 or 5 months now.
You miss her, Simon?
Absolutely. Yeah. Regular Zoom calls, but it's not quite the same.
Yeah.
No, it's not at all. And Simon, you go in regularly?
Yeah, I've actually been going in most days, to be honest, throughout. If I have some things to do from home, then I will. But I have sort of an office where I can isolate myself, so it's been pretty easy for me. And I only live down the road as well, so I can get here without public transport or anything like that.
So, and Simon, has the whole lab sort of disbanded given the pandemic, or are there still a few?
Initially, yeah, but now we're sort of starting to ramp up again now that sort of non-COVID research is also starting to come back online. Most of our lab staff are now back, though I think like everyone else, if they're doing anything that doesn't require them to be at the bench, then they tend to work from home. But yeah, so most people now are sort of back or on their way back.
How about you, Heather? Do you have plans already to return, or are you going to hang out in Ohio for the time being?
Yeah, well, I just got my teaching assignment from Columbia, and the class that I'm TAing is going to be online only. So I guess it's really, really up to me, but I'm so much more comfortable in Ohio. Like, there's just so much more space. My mom and I put in a vegetable garden this year, and that's been so much fun. Like, all these things that I just could not do in New York City. So, we'll see.
A pandemic garden.
Yeah.
That garden is dangerous, though. You also broke your arm because of it.
Oh, yes,
that wouldn't have happened in New York City.
Yeah.
You broke your arm in the garden? What were you doing?
Well, it wasn't in the garden. It was on the back deck on the way out to the garden. But I was so excited because our seedlings had just germinated. They were like this big. So I'm kind of like trucking it down these old wooden wet steps on the deck, and just feet flew out from under me, and I caught myself with my shoulder, but like the rest of me kept going, and my arm just kind of like went up, and I actually broke my shoulder. My humerus just snapped. So
wow, is it healed now?
yes, I've been in PT for like 3 months, but the bone is like completely healed. Everything's fine.
Yeah. All right. Good.
But yeah, dangerous garden.
Well, I hear there's good medical care at Columbia, so you probably want to hurry back and get back to the lab as soon as possible.
Yeah, there is. There's good medical care. So let's before we talk about your science, let's get a little history of you. So Simon, where are you from originally?
Originally from England, as you can probably tell. Yeah, so I actually been in the US for 13 years now. I sort of finished all my education over in England.
What part of England are you from? Are you not from Yorkshire, are you?
I'm not from Yorkshire, no. I'm not. I'm from the south.
South?
From Kent.
Okay.
So south of London, right down on the coast. Yeah, so Kent.
And you went to college in the UK?
I did. I did my undergrad at the University of Wales in zoology, and then from there actually, I was going to become a zookeeper. So I when I graduated, I was offered a job as the trainee elephant keeper at London Zoo, which I thought was an awesome job, awesome opportunity. And then literally the very next day, unfortunately, the elephant keeper was killed by the elephant.
Wow.
At London Zoo. Yeah, really bad. And so, of course, my grandmother was right on the phone immediately, making sure that I was not about to go and take this job working in this very dangerous place. And London Zoo, of course, then decided that London was no place for elephants and moved them outside of the city up to their wild animal park and offered to put me in the aquarium instead. And while it was probably short-sighted of me, I decided that actually cleaning fish tanks was not quite as cool as working with elephants. And so decided not to do that. And actually instead went to do a temporary position at the Institute for Animal Health, or what is now known as the Pirbright Institute. Because at the time there was a foot and mouth outbreak that had just started in the UK. And so I went to sort of be one of the technicians doing diagnostic testing and really discovered that I loved the lab side of things. I loved working with viruses. I sort of still had that animal health component, but it was more from a research perspective. And so after doing that for a couple of years, went and did a PhD in animal virology. Focused on sort of a episodic hemorrhagic disease virus. It just rolls right off the tongue. And yeah, so I did that. That was both at Oxford and also the Pirbright Institute. And what's nice about that virus, actually, in Europe, that's really regarded as a sort of a virus of livestock, of agriculture. But in the US, it's very much a virus of wildlife. And so using that same virus, I was sort of able to pivot and market myself as a wildlife virologist, and then went to San Diego Zoo in California to join their wildlife disease department. Really fantastic opportunity. I mean, some of the world's best animal pathologists that are based there. And so I sort of then switched and started working on all sorts of different things, really. That's where I got into sort of viral discovery, and that whole thing. And then after a couple of years there, really realized that humans were a big part of all of this. And that's where I sort of really, learned to, appreciate the One Health idea. And that was when USAID had really started this program called Predict. And so I then moved here to Columbia to be part of that and sort of seal up that One Health triangle, if you like, and sort of now think about viruses coming from wildlife and how they actually then spill over into people. And that's what we do now.
When you were at Pirbright, did you know Dave Rowlands?
I did not, no.
But do you know the name? He was an old foot and mouth guy. Yeah. Because I'm basically a picornavirus person, so I used to see all the foot and mouth people. We knew all about Pirbright. I think there was an outbreak where the sewage was faulty or something like that. Is that right? Something coming out of that.
Yeah, it was. And so there's a next door. There's a pharmaceutical company. and then there's Pirbright. They were also on the same site, and it was because of a leaky pipe, essentially. That still don't really know how it happened, but a leaky pipe, and the virus got out somehow through that pipe. Yeah.
So, and Simon, how long have you been at Columbia running your group?
10 years now. Yeah, so 3 years in San Diego, 3 years doing the postdoc there, and then 10 years now here at Columbia.
So you came to the CII initially, right?
Yeah, I joined initially as a postdoc, and then from there I sort of just went up the ladder to associate research scientist, and now to assistant professor.
Cool. So Heather, you're from Ohio, right?
Yes, I'm from Ohio originally, but I did my undergraduate work in Nashville, Tennessee at Vanderbilt. And like Simon, my initial intentions for my life were quite different than where I've ended up. I originally wanted to do veterinary medicine. So, still kind of like in the animal realm and in the health realm, but like a completely different world than where I've ended up. But I first got my hands into lab work at Vanderbilt in a chytrid lab, so in an amphibian chytridiomycosis lab with Louise Rollins Smith. And she's the one who kind of like opened my eyes to this whole world of diseases that can like transmit between animals and humans. And like I had no idea that was a thing at the time, but it just blew my mind. And I remember reading this article like in the New York Times. I think it's probably almost 10 years old at this point, but it was about zoonoses and spillover. And I went back and read it recently, and actually, Simon, you're quoted in that article. So like our paths had crossed before I even knew.
Hopefully nothing horrible.
No, it was all good stuff. But yeah, so after that, I kind of had decided that infectious disease was really what I wanted to focus on. But as you well know, there's so many facets to this kind of work. There's like the ecological and animal side. There's the human and epidemiology side. There's just so many different directions that you could go when you want to further your education in a field like this. But what I decided to do was my master's in public health in microbial disease. And so I ended up at Yale and was doing a 2-year program. And then as part of that program, you have to do a summer internship in between the first and second years. And that's how I ended up with Simon. So I just, sent him a cold email and told him that I thought his work was super cool. And he emailed me back, and then the rest is history. So, finished my master's in 2016 and then moved to New York City to start working with Simon as a technician. And then started my PhD in 2018. And so I'm going into my 3rd year of my PhD at Columbia, but not in epidemiology. Now I have switched to ecology and evolution. So kind of like getting the rest of the educational kind of like foothold that I need to really feel like I'm approaching these questions from multiple directions. And so it's been really, I think, a good opportunity to have all of those varying educational experiences and to kind of like bring all these things together in my own work. So that's how I've ended up here.
You mentioned that you're teaching a course this fall, I guess. What is that? What course is it?
I'm TAing, so I'm not technically teaching it, but I'm TAing the Fundamentals of Evolution course in E3B.
Who teaches that?
Darren Eaton.
Okay.
Vincent, have you taken a Fundamentals in Evolution course in your?
I never did. No, when I went to college, which is when did I go to college? 75 to 79. Is that right? Yeah, I think so.
I don't know. Those are big numbers. A long time.
Evolution was very different, as we've talked about on this show. It was pre-molecular era, and it was, I think, not as interesting. It was mostly cataloging, right? But it's all changed. So I would love to do that now. I should, but
Yeah, no, the genomic era has really changed the conversation. But I think what you're saying, Heather, I mean, I'm obviously kind of preaching to the choir, but shifting gears into the ecology and evolution as sort of a focus, kind of a foundation, makes a ton of sense, because then you can build off of that and all kinds of kind of return and move out again as you're moving into the next step. So that's really exciting.
Yeah, it was a bit difficult to convince the department, I think, at times that, someone coming from a human health and epidemiology background was a good fit for an ecology and evolution department. But I think, like, in the end, it's all made sense, and it's been good for everyone. So.
Yeah, I can't think of a better fit, actually. We need the zookeepers, the elephant keepers, the epidemiologists, all coming together. This is what's exciting about the field of evolution these days.
Heather actually is someone that that I think is of growing importance, with students nowadays. Because Heather's someone, of course, that understands very much the lab side, working in a lab, but also is able to sort of incorporate the theory and the practice from completely different fields like epidemiology and evolution and ecology, and synthesize all of those together when addressing some of these questions. And it's important because many of the questions that we're interested in are not easy. They're not easy, and there's no single discipline answer or single discipline approach that can really be taken to sort of make any headway on them.
Yeah, agreed. And I guess that's kind of captured in your lab's work, Simon. So the ecology and evolutionary drivers of virus spillover and disease emergence, kind of grabbing from a lot of these disciplines. It sounds like that's exactly the kind of core of your own work.
Yeah, I mean, I think so. So for us, it's really about trying to understand more about the determinants of zoonotic risk, and then sort of incorporating these ecological and evolutionary ideas and trying to understand how those processes actually shape that risk. So in my group, we conduct surveys for undiscovered viruses in wildlife. And then we try and identify the ones that we think have some ability to infect people, that they may have some of the necessary genetic prerequisites for human infection. And then also we like to study how they evolve that ability in the first place. So what are the underlying evolutionary mechanisms that sort of mean that some viruses have this ability and some don't, or as we'll be talking about today, that can alter those traits. And then also, what are the ecological influences on those mechanisms too? Things like host adaptation. And so I think, we like to sort of have this sort of very broad framework for thinking about zoonotic risk and trying to understand not just what are the risky viruses that are out there on sort of an individual virus by virus basis, but what are those broader mechanisms and processes that shape that potential in the first place?
Yeah, Heather, you mentioned that you didn't know years ago that human viruses could come from animals. Well, today most people don't know that. They're amazed that we get viruses from bats. They go, "How could that be? How could that be?" They say.
When I told my parents that I was going to study bat viruses, they were just scratching their heads like, why would you choose a career like this? And if I would tell people what I do, they would just, what are you talking about? Bats and viruses and the blood and pee? Like, why are you doing that in the lab? And and so I was that weird bat girl. Like, even in my department, it's like, okay, she does something with viruses and bats. But now everybody everywhere knows what a coronavirus is. And it's just really I'm so cool now. It's a completely different reaction when I tell people what I do. So yeah, you're right. I'm very familiar with that confusion.
Yeah, you're just ahead of your time.
Yes, I call myself a coronavirus hipster now. So I liked them before they were cool.
Well, it's Simon, listening to you, the thing is, we have to use the information that people like you get, and we didn't. That's why we're stuck in this situation, right? After SARS-1, we accumulated a lot of interesting stuff about pandemic potential, and we didn't do anything. And now we're caught shorthanded. So I hope next time it'll be different, but I don't have a lot of faith. And the problem is we don't pay much attention to things when they're not an issue, right?
Right. It's true. And it goes back to what I was saying earlier too, about these being extremely large, challenging problems. You can study the drivers of emergence for 10 years and you get to the end of it and many will expect you to have found the answer or the solution. Okay, well, how do we predict what's next, or how do we stop this? And the reality is it's an extremely, extremely difficult thing to do, and it'll take decades of research before we can do that. I think that anything that is as complex of a problem as zoonotic emergence is going to take lots and lots of different people coming at this from lots of different perspectives, and it's going to take a really long time.
What's really long in your opinion?
Well, if I'm talking to a funding agency, it's going to be at least another 10 years. It has to be long enough that I'm secure with money for a while, but not too long that they don't think it's worth it.
Yeah, I asked Mark Dennison once, just a few months ago, I asked him, who's at Vanderbilt. Did you know Mark Dennison, Heather, at all? Do you know the name?
I think he's in the medical school, right?
Yeah, he's in the medical school.
Yeah,
that's right.
I didn't know.
Okay. Anyway, I said, could we have had an antiviral, a pan-coronavirus antiviral after SARS-1? He said, yeah, just fund me for life. I would have had it for you. That's the bottom line.
It really is striking. And I think just to underline what you're saying, Simon, so this bringing together the different fields, this is sort of actually one of our sort of motivations for running this podcast, TWiEVO, is this opportunity for trying to enhance those collisions or support those collisions of coming at things from different expertise, because this feels, I think, like the perfect storm from an ecological, evolutionary, epidemiologic, health, the medical school, sort of these conversations now across campus where all of a sudden the ecologists and the pediatricians like Mark Dennison are comparing notes in a real way. And so how can we support and sort of continue to motivate those conversations? And I think, Heather, this kind of cross-training along these lines, it can be both at times frustrating, that kind of, how do I, jack of all trades, master of none? What am I, the expert?
Yep. Yeah.
But at the same time, I think it really is worth it to hang in. And because no one else has this combination of unique sort of experiences and background. So,
yeah, thanks.
So I want to talk about this. I guess it's still a bioarchive. Must be in review, right? Evolutionary history of ACE2 usage within the coronavirus subgenus Sarbecovirus. It's still being reviewed. Okay.
Yeah, exactly. Yeah.
And Heather, you're the first author.
Indeed.
And Simon, you're the last author. So maybe, Heather, you tell us how you got involved with this project.
Sure. Yeah. So Simon and I have been percolating with coronavirus type ideas for quite a number of years now. And so when the news kind of broke that there was a novel coronavirus in China, obviously we were on it like night and day, checking the news all the time, always on virological, and just trying to keep up with everything that came out. And so all this news is coming out, and everyone's kind of trying to put it together. Where did this come from? What does it mean? And, we learned that this virus, it's, SARS related, but within the Sarbeco group, it's about as distant from SARS as you can get. So, relative to all coronaviruses, it's closely related to SARS-1. But relative to this subgenus in particular, it's way out there in space in a place that we didn't even know viruses were phylogenetically. So, this information is coming out, but then, this information comes out that somehow this virus is using the same receptor as SARS-1. They're both using ACE2. When we know already that there are a bunch of intermediate viruses phylogenetically that aren't using ACE2, and they have these large deletions in the receptor binding domain that are most likely responsible for rendering them incapable of using ACE2, or human ACE2 at least, I should say. And so this was a really interesting question to me, and I think to a lot of people, on how you could get this pattern of two viruses that are so far apart within this little group Sarbecovirus, yet they both have the trait of ACE2 usage. And so, that's kind of how we got into this, into the weeds of really like putting all of this information together and trying to figure out how you could explain this. And other people were interested in this question too. I think really early on, the idea of recombination was kind of like a hot topic, and people were saying, yes, there's recombination. No, there's no recombination. And, if you run these things through recombination detection programs that, there's a bunch of them, they won't flag recombination between SARS-1 and SARS-2. It doesn't come up. And so, everyone kind of settled like, okay, well, there's no recombination. And then the convergent evolution hypothesis kind of like is really popular with some people. And so there's like these two competing ideas of, did the trait arise through recombination, or did the trait arise a second time through convergent evolution because of some sort of selective pressure on this other group? So that's really the conundrum that we set out to investigate with this study.
And the trait you're describing, this is the ability to recognize the human ACE2 receptor. This has been, we've talked about this on TWiEVO, and of course on TWiV, this has also been a massive area of conversation. And then maybe just really quickly, this notion of an intermediate, phylogenetically intermediate, maybe just help me out with that a little bit.
Sure, totally.
So the two possibilities would be that you're used to have the ability to bind ACE2 and you've let go of it, or you've not had that and you might be on the way to gaining it. Help us to understand that a little bit.
Yeah, so you've basically nailed it. There's really two directions that it could go. Either you have an ancestral group of Sarbecoviruses that is not using ACE2, and as they evolve through time, somewhere along the way, there's a mutation, an insertion, a deletion, some kind of selective pressure where a group of viruses gains the ability to use ACE2. And then you'll have a phylogeny where you have a bunch of viruses at the base of your tree that are not using ACE2. And then you'll have a chunk, like a little group that are using ACE2 because they evolved that trait somewhere along the way. And so you could also have it the opposite. You could have an ancestral group that's using ACE2 and then viruses that evolved from that group that subsequently lost the ability. But what we see here is we see this SARS-2 group is actually ancestral on the tree. And then you have the other Sarbecoviruses kind of evolving from that group, not necessarily directly, but like from a common ancestor that was like before the split, if that makes sense. And so you have the ancestral SARS-2 group using ACE2, then you have viruses evolving that are from more recent common ancestors that do not use ACE2, and then all of a sudden you have SARS-1, which pops up within this group of non-ACE2 users, and it's also using ACE2. And so that's the conundrum. Like, how do you get that pattern? How does that happen? And that's why, there's only two explanations. Well, there's three, but the two that we address in the paper are recombination. So either SARS-1 or SARS-2 were able to gain that trait by recombining with each other, or convergent evolution, where there was some kind of selective pressure. So there's some reason that ACE2 usage is really advantageous for these viruses. And so they both converge onto the same phenotype, essentially. And then the third hypothesis, which we didn't address in the paper, but which has been brought up by a reviewer and a colleague, would be the hypothesis of persistence, basically. So if you had an ACE2 using virus that had been ACE2 using all along and it had somehow persisted despite all of its nearest neighbors having deletions, which is not a parsimonious argument whatsoever, but is possible. Yeah.
Sounds like you're dealing with some reviewer comments there. Simon, can I ask you to set the stage for us a little bit? So some background on what are these Sarbecoviruses? What does that sort of encompass? And then where did these new viruses that you've kind of added to the sample here? How did you come about those?
Go
ahead, Simon.
Yeah, no problem. So obviously the Sarbecoviruses are sort of a subgroup within the coronaviruses that include SARS-1 that emerged back in 2003. And after that, of course, that stimulated a whole bunch of surveillance in wildlife, particularly in bats, and many related viruses were then found circulating in bats. Initially, those viruses did not have the ability to use ACE2, but over time, they also found variants of these Sarbecoviruses in bats that could actually use ACE2. And then, of course, most recently, we have another one also classified within the Sarbecovirus subgenus, which is obviously the agent that is responsible for the current pandemic. Now, the new viruses that we found were actually part of the USA Predict program that Heather and I have been part of for the past several years. And the goal of that project really is to sort of try and build a better global capacity, if you like, to be more prepared for these sorts of events. And that can mean anything and everything from training local wildlife biologists how to sample bats safely and get those samples into a lab, and then training local lab technicians and lab staff how to use discovery and detection type approaches to find novel viruses, right the way up to policy and building connections between government ministries to sort of really facilitate a flow of information in sort of very much, again, a One Health paradigm. So as part of that program, as part of Predict, obviously there was surveillance going on in about 30 different countries. Uganda and Rwanda were two of the countries that were part of that program. And as part of those efforts, we found two novel Sarbecoviruses, three strains, but really two viruses, one from Uganda and one from Rwanda. And what's interesting is about these viruses is, of course, they provide some extra context. They provide some extra clarity that we didn't have previously. As you mentioned, what they do is they serve as these intermediate viruses, and they're intermediate because they have some of the things along that multi-step process that Heather was talking about between being an ACE2 user and not being an ACE2 user. These viruses from Africa seem to have some of those features from the ACE2 using group and some of the features from the non-ACE2 using group. And so they're sort of like halfway between the two on this journey to either usage or not usage, depending on which way around you're looking for it. So it can be very helpful. It shows, obviously, that these Sarbecoviruses, they're found outside of South and Southeast Asia. So obviously, we knew about some divergent strains in Europe too, but we're showing immediately that actually the distribution of these viruses is actually quite broad, much more broad than we initially thought. We also now know that these viruses don't use ACE2, and so potentially do not represent the same level of threat as something like SARS-1 or SARS-2. Though I should, of course, clarify that we don't know what receptor they do use, so it doesn't mean that they're not zoonotic, but it just means that they're probably not zoonotic via ACE2. And so we start to be able to do lots of things, right? I mean, we start to be able to delineate the geographic boundaries of where we find ACE2 using viruses. And we start to delineate the phylogenetic boundaries between what is an ACE2 competent spike and not competent spike. And that, of course, means that when we find new things, we can very quickly predict whether or not it's potential for human infection or not.
So when you say, bind ACE2, you're talking about human ACE2.
Human ACE2.
Bat viruses that bind human ACE2, do they also bind bat ACE2? And do they enter via bat ACE2?
There's not actually an awful lot known about that, but I think it's a really important avenue for research. The one thing we do know is that the ACE2, especially that the region that is involved in binding, is really quite highly conserved. And we know that substitutions at that interface will certainly have an impact on the affinity of that interaction. So some ACE2s will be better binders than others, for example. But there hasn't been a lot of work characterizing the sort of the full extent of interactions between all of these different viruses and a whole panel of different bat ACE2s to evaluate what is a good binder, what's a bad binder, and what is can't bind at all. But I do think that's a really important point. What we do know is that for the bat viruses, that are non-ACE2 users, they probably don't use bat ACE2 either. And the reason we think that, but don't know that, the reason we think it is that the sequence, the genetic sequence in those key regions of the virus are very, very different. So structurally, it would be completely incompatible. And it would be a large surprise, let's put it that way, if we found out that they did use bat ACE2. On the contrary, the African viruses are interesting because the structure of the receptor binding domain in those viruses actually is similar to ACE2 competent strains. And what's different there is the actual sequence itself. And so in that case, we think it's more about having very, very poor affinity for ACE2, or at least for human ACE2. And there, in that case, it would be particularly important to verify whether or not those viruses actually do use bat ACE2, just not human ACE2.
So, but bat ACE2 is different, and so that begs the question, how the hell does a high affinity human ACE2 virus emerge? Is it just random?
I think...
Well, yeah, Heather, you should take this, and your sort of ideas on contingency, I think, are really great.
Contingency, yeah. Well, I wanted to clarify that the differences between human ACE2 and bat ACE2 are very, very small genetically. So there's only a handful of substitutions. So the idea that we might by chance randomly get a high affinity human ACE2 binder just by, the random process of the evolutionary arms race and the generation of mutations in order to escape the bat immune system, these processes that generate new variants could very easily have generated a variant that just happened to have the correct combination of residues that just made the binding with human ACE2 that much better.
Right. It would not have. It should not affect the bat ACE2 binding, right? Otherwise, it would be selected against, I guess. Right?
Exactly. Yep. So that's the key: is that these viruses are competent at binding bat ACE2 and human ACE2, and there's varying gradients of affinity, as Simon was saying, for binding the bat ACE2 and for binding the human ACE2. But that evolutionary space, if you will, is very much overlapping for the bat and the human binding.
Yeah.
So there's no virus out there evolving to be a better human pathogen.
Right. That's why these are random events. But as you say, if bat ACE2 is already close, then just randomly it could.
Right. And it can go the other way. So we work with Ebola viruses too, and we found a novel Ebola virus in West Africa, which it was found in a bat living inside people's houses, like multiple bats living inside people's houses. And so, of course, immediately you'll think, oh my God, thinking about what we know about Ebola viruses. And so we then did, with Kartik Chandran's group, we did some experimental work to show that actually this new virus, which we called Bombalii virus, does have the ability to actually bind and enter human cells. But what was interesting about it is it didn't do it as well as Ebola virus does, the pathogenic Ebola virus. When you actually compare them, the pathogenic Ebola virus is really good at getting into human cells, and this new virus that we found is actually not so good. And when we looked into it a bit further to try and understand why that is the case, we found a single mutation in this new virus that actually is responsible for reduced entry into human cells. And when you evaluate how well that new virus does in cells of its natural host, of the bat host, we also learned that that one mutation makes it really, really good at entering those cells. So here we have a situation where we have a virus that has evolved for optimal entry into its natural host, and it actually reduced the zoonotic potential in humans. So it can go both ways.
Well, it makes perfect sense because as you say, human infection is not the goal. The natural host is the bat.
Right.
Mm-hmm. The virus has no idea. It's not
of
conscious. Like, it's not doing anything on purpose. And this really, I think Simon was hinting at this idea of contingency. And it really just points out that for every situation like this, it really just depends. It really just depends on what kind of tools the virus has to work with and like what kind of genetic structure it has. It's just like how humans, we can't just evolve wings because it would be really useful to have wings. We don't have the genetics on which to evolve wings. So virus evolution leading towards, zoonotic spillover or anything like that is contingent on what that virus has. So if the viruses have the ability to be using ACE2, then that just means that, that they could potentially become using human ACE2 as well. It's like, it's just the way that it happened. It's just contingent on what the virus had before. It's not like anything was happening on purpose or like for any particular goal, which I think is hard for us to remember sometimes. But yeah, I think that the Ebola story is a really good example of that as well. Is it just depends on what the situation is for every different viral family. It's going to be different.
Yeah. And just to connect the dots a little bit on Kartik Chandran, you can actually hear him talking about another collaboration with Sarah Sawyer on bat virus evolution. I think that's TWiEVO episode number 2 in our early days, talking about straw-colored fruit bats. And a related story there. And speaking of that, so Sarah Sawyer and Mike Farzan did some work looking at ACE2 evolution among some bat species, where it does look, I think I heard the phrase evolutionary arms race as a sort of simplified analogy for these recurrent genetic conflicts, that there is real action in ACE2 among bats, whereas in primates, including humans, actually a much quieter history, meaning that ACE2 is almost identical if you pull it out of our closest relatives, the living great apes, the old and new world monkeys. But this kind of does raise the question for the viruses in particular, the ones that you're seeing from Africa. I think you noted that you're not actually sure who the bat is or the host is. Is there any possibility of making progress on that front?
Well, so in addition to the millions of dollars that we need for sort of doing the surveys and understanding zoonotic emergence, we also need to support those long-trained taxonomists that really we depend on for this kind of for exactly this kind of thing. Actually, bat taxonomy in Africa is not well understood. We're constantly getting updated and refinements to that. We actually found, again, in West Africa, again with Ebola viruses, sorry to go there, I know we're talking about coronaviruses, but there we found a bat that was positive for what we think is actually the West African outbreak strain of Ebola. And it was a similar thing, right? It was a bat that was positive, but we struggled to identify what it actually was. And just coincidentally, there was a bat taxonomist that was working on bats in that region that had just classified this particular species. And we just happened to bump into each other at a conference. And so he was able to help us narrow it down. But no, the short answer to your question is no, we haven't made any headway. And so what we do is we obviously generate the genetic data and submit it to GenBank. So that at the very least, we have a genetic barcode of what that species is, even though it doesn't actually have a name yet. Mm-hmm. Same family, though, of course, as all of the other Sarbecoviruses.
So this makes it very clear that we need to do more sampling of wildlife. And so it makes even crazier the withdrawal of NIH support for EcoHealth Alliance's work in China, right?
It really does. Because the one thing I really like about this study is the context that it provides, right? I mean, this study it's not the vaccine, which is what we really, really want. But it is so useful. These natural history studies where they sort of try and make sense about the evolutionary journey that these viruses took to be what they are today, and the sort of the processes that they went through, are completely contingent on us going out and surveying wildlife and identifying these viruses in the first place. And even if 99.99% of them turn out not to be human pathogens, which is probably actually quite true, it doesn't matter, because we learn a lot about the counterfactual, as we call it, in that sense. We learn a lot about the ones that don't. That tells us a lot about, obviously, the ones that do. And it really feeds these natural history studies that tell us more about just how these viruses came to be.
Aside from Asia and now your isolates in Africa, are there Sarbecoviruses in South America? You said Europe there were some, right?
Europe, there are not found by our group, but there are two. Is that correct, Heather? 2 or 3 sequences from Europe?
There is one genome. So you'll see one genome that we reference in the manuscript, but there are actually quite a few PCR fragments. So the way that people typically search for viruses in these wildlife samples is through consensus PCR, which can broadly bind to different types of coronaviruses in samples, and it targets a very short region of the polymerase gene. So there are a bunch of these short little polymerase fragments in GenBank that are Sarbecoviruses that are from Europe, from France, from Italy, from Spain, from various places across Europe. And but we just don't have full genome sequences for those.
And none from North or South America?
As far as I'm aware, there's no Sarbecoviruses in North or South America. And my, suspicion for why that would be the case is just because their natural host is not found there. There are no rhinolophid bats in the Americas.
We hope that holds, because we published a paper in Virus Evolution looking at global patterns in coronavirus diversity and predicting hotspots of where we would find Sarbecoviruses, and we did not predict any hotspots in the Americas. So we're hoping that holds.
Well, and that's where the differences in ACE2, which are already evident from sort of limited sampling of bats, could play into the favor of North and South American bats if their ACE2s have diverged, at least for the ACE2 users in that case. So I'm just looking at figure 1 in the bioRxiv manuscript, and it's this really nice phylogenetic tree, and then linking that up to the geography of where the viruses were located from their bat hosts. And it looks like, all of the ones in Southeast Asia, sort of the now famous lineages 1, 2, and 3, and then sort of separate from there, diverged, or as you're saying, intermediate in terms of some of the traits, are the African. But then also, it's sort of, my European geography is a little rusty, but I think in Bulgaria you have a link out there which is actually highly closely related to the African bats. I'm just curious, what do you make of that? How is that? Is there some exchange going on here, or what's happening?
So that one genome that you just mentioned, the one from Bulgaria, I think it's like BM48, is the only full genome that we have from Europe. So that's the only one that I was able to include in this tree. However, if you include in this tree the other small fragments from Europe, as I was mentioning, they all cluster exactly with the one from Bulgaria. So there's a distinct cluster that's found in Europe, and then there's a distinct cluster that's found in Africa. And they just happen to be sister to each other. But as far as I'm aware, and just according to the phylogenetics, there doesn't appear to be any mixing or crossover between those two regions. But of course, I could be proven wrong at any moment. But given what I've observed in the phylogenetics so far, there's very distinct geographic clusters of Sarbecoviruses in Europe and in Africa. And they're similar to each other, more similar to each other than they are to the Asian ones, but they're distinct from each other still.
I think they look closely related because there's so few of them known. But as soon as we start, filling out that tree with more and more genomes, they're not actually going to look quite as closely related as they do right now.
This is that point of context that you already mentioned, Simon. The more sampling you do, the better sort of picture that emerges about the relatedness of these things.
Right, right.
Yep. So, and Heather, you mentioned recombination being sort of a central mechanism for the diversification of coronaviruses. And so maybe walk us through how you sort of took that view as you started to analyze and compare massive differences in phylogenetic relatedness, depending on which parts of the genome you put together for phylogenetic trees.
So recombination in coronaviruses is something that Simon and I have been very interested for quite some time. And there's a lot of literature on recombination in the spike gene, and it just kind of naturally leads to this hypothesis that when coronaviruses recombine in the spike, and the spike obviously contains the receptor binding domain, which is responsible for binding host cells. So the spike gene determines which hosts you can possibly infect. So when coronaviruses recombine and they swap out spike proteins or regions of the spike protein, then there's this hypothesis that allows them to subsequently infect other hosts. And it could also be a mechanism by which viruses swap spike proteins and then potentially gain the ability to infect human hosts. So this process of recombination in coronaviruses, all coronaviruses, not just Sarbecoviruses, is something that we've been quite interested in for a number of years now. And so when SARS-2 and information about SARS-2 started popping up, we were right on it. Like, is it a recombinant? We need to know right now. And that was the first thing I did when the genome count came out in January. I'm running recombination detection algorithms, and actually was a bit disappointed initially to find out that there's not a lot of signature of recombination in the SARS-2 genome. And so, I felt a little defeated because that didn't necessarily help our hypothesis that recombination drives spillover. But, the hypothesis is not that recombination is necessary for spillover. So it's still okay. And we're still going to investigate this, I was thinking. So I'm trucking along, making my phylogenies and, looking at the spike gene, looking at the receptor binding domain within the spike gene, comparing these phylogenies for different regions and noticing this pattern where, the polymerase tree, which is really nicely outlined in figure 1 and has everything kind of broken down really neatly, really kind of mixes up when you look at it in the receptor binding domain. So you still have the geographic breakdown for the most part, but what you see is the SARS-1 clade, which used to be up within lineage 1 in figure 1, has now completely changed positions and is now down and is sister to the SARS-2 group. And so that was really striking. So this nice pattern of phylogeny that we saw before was completely not flip-flopped, but rearranged in the receptor binding domain. And so that, to me, was a signal of recombination. So when you study recombination, when you learn to look for these things, those big jumps in phylogeny are usually your first clue that recombination has happened. And because everything else in the tree stayed in the same place relatively between the polymerase and between the receptor binding domain, and the only thing that really switched places was SARS-1, and then there's a couple of bat viruses that are really close to SARS-1 that also switched with it. Because those are the only things that really significantly changed position, that kind of led to the hypothesis that SARS-1 group was a recombinant group with something related to SARS-2. So not recombinant with SARS-2, but recombinant with something that must have been related to SARS-2 at some time in the past. So this was really exciting because naturally SARS-1 came first and SARS-2 came second. So your brain wants to assume that SARS-2 is the recombinant. But this idea that possibly SARS-1 was the recombinant and it happened quite a while ago was actually a really interesting idea, and I think was just a different way of thinking about it that we hadn't been thinking about it before. So that's kind of how we got onto this trail of recombination and started investigating this hypothesis. Does that
and highlights that the potential for emergence is one piece of the puzzle. That there's a whole there's a whole likelihood component that goes into spillover and emergence too. And, you have to come into contact with that bat at the same time that the bat happens to be infected with enough virus that it can be transmitted from, into a person or into an intermediate. So there's probably a whole suite of viruses out there that have a similar potential and never will because they simply don't have the opportunity.
One thing that always strikes me in these discussions is that, the receptor binding is obviously a key step because it's first. And if that doesn't happen, nothing else does. But all the steps downstream have to be compatible between bats and humans, right? And we don't talk about that much because I guess we don't have a handle on them like we have a handle on ACE2 spike, right?
Right, and because the problem's again just so big. Obviously, the fact that a virus is going to be a human pathogen, not only does it have to go through this whole sort of evolutionary process of having the ability to get into a human cell in the first place, but whether or not disease manifests from that is the result of hundreds and hundreds of protein-protein interactions between the virus and obviously the host cell and surrounding cells. But that's a really, really difficult thing to measure. And so you can take certain traits that you think are important, cell entry, of course, being one of them, interferon being another one. You can look at whether or not these viruses really have any ability to antagonize interferon, and potentially some sort of replication type thing to think about, whether there's any sort of host restriction going on. So there are things that you can do in a reductionist way that help you understand whether or not you elevate this particular virus in your concern meter. But none of them are, of course, going to prove that it is going to cause disease. And of course, none of them will prove that it will. And that also goes for if you have the virus. We as virologists we like to have the virus. We like to work with the virus. But even then, we have problems. Again, harping back to Ebola viruses, you have Reston virus, one of the Ebola viruses, which does cause disease in non-human primates, but does not cause disease in humans. And so there's no simple solution to this, none whatsoever. And so you sort of arrive at this point where you think, okay, well, shall I sit back and do nothing, or shall I try and understand these things as best I can?
You have to have model systems, obviously, because nothing's perfect.
Yeah.
And I think, one approach is simply to ask, will viruses, if you can get viruses from bats, will they infect ACE2 producing human lung cells? And Ralph Barrick asked, will they infect ACE2 transgenic mice and cause some kind of disease? I mean, they're not perfect, as you say, but you don't want to sit back and do nothing. That's not what we do.
Right.
Well, but it does really illustrate the complexity here, right? It's kind of contingency on top of contingency on top of, as you kind of peel back the layers of the onion of the replication cycle and then the immune response. And so, by the old analogy, the drunk looking under the spotlight for the car keys, makes sense that we look at ACE2, the receptor where we can do the biology. The assays are in place, including some that you did in the preprint to back up sort of some of the function here that you're predicting from the phylogenetic distribution.
Right, exactly. Yeah, work. So that's Michael Letgo's work, of course. Yeah, so fantastic.
Yep. And, again, illustrating sort of putting together from field collection all the way to the model system to try to understand the function here. But it really, I think to me, also highlights just the massive numbers at play, right? So because it's not just, do you recognize the human receptor in a way that's productive, but then, can you still replicate? At the same time, it doesn't have to be perfect, as is already mentioned, because, and I think we're seeing this in the clinic, that in fact, in some cases, the virus is not sort of like going in with a scalpel and disarming the immune system, but in fact, the immune system is panicking. And some of the symptoms related to that are emerging from that mismatch as you start to see this happening. And so these are the collisions, right? As humans become increasingly closer to wildlife or destroying habitats, now the numbers of the times that you are exposing diverse viruses to human cells that might be vulnerable in some way. I mean, it's really, I think, hard to grasp. It's kind of mind-boggling how many of these interactions are actually happening to sort of thread these multiple contingencies.
I agree. And I think about actually some of Peter's work in China, where they were sort of doing serosurveys of people and showing that there are there are people with clear evidence of exposure to SARS-like viruses. Exactly what the virus is of course not known, but clearly something within that Sarbecovirus subgenus. And I think that alone shows that there are interfaces right now that at least allow this sort of spillover to occur.
So this brings up the topic of other than bats. When the cat crawled on your lap, this reminded me of other animals. So bats are clearly a worry, but what else? Are rodents another worry for zoonotic events?
Yes, I mean, I think so. They are. I hesitate because I don't want to sort of be alarmist and sort of start stigmatizing entire groups of animals. People talk a lot about bats as being special, for example, and they may be, but let's not forget that bats are also the second most speciose taxa on the planet. And so when we say bats carry a lot of viruses, well, of course they do, because there's over 1,400 species of them. So it's absolutely, or should be, no surprise to anyone that they carry a lot of viruses. And so the same thing will go for rodents, because they're even more speciose than bats. And so, yes, they too will have many viruses that have the potential to spill over into people too. And so surveillance of rodents is also important. It's also very difficult. Catching rodents out in the forest is not an easy thing to do.
It's harder than bats.
But, as someone that goes to the field once in a while, I will make that qualification before I make a call on how easy or difficult they are for the people that are actually out there having to do this work. But yes, they, as much as I understand, rodents are pretty tricky. It's not so obviously. There are studies of New York City mice and things like that have happened here at the center, and it's not the same thing as going to a boiler room at the bottom of a building and collecting 50 mice. It's a whole different ball game.
So returning to the manuscript, and you start to drill in. So you've got these big this big view of the relationships and the difference between so I think you mentioned the polymerase. This is the RNase dependent RNA-dependent RNA polymerase, RDRP, which is, I guess you would assume, or I think you say this in the manuscript, you would assume that there has been less recombination here. I guess that would just be an output of the fact that this thing is so conserved. This is sort of like a non-negotiable part of the replication cycle for the virus that there might. So recombination, I would assume, might be happening just as much, but the natural selection might not have as much of a foothold to, whereas the receptor binding domain, which you mentioned in the sort of shuffling of the tree, that part of the encoded in the gene for the spike protein. This is again how you recognize the ACE2 receptor, where there's more action there. And so I guess the idea is this correct, that recombination would be happening just as much, but that you'd be selecting for these differences. Does that make sense?
Yeah, it's possible. That's a really interesting hypothesis that also Simon and I have kind of gone back and forth on, is whether recombination is equally probable or possible anywhere in the genome, and just that there are certain parts of the genome that are more easily swapped out than others. And so recombination within the polymerase might render that virus no longer capable of replicating, whereas recombination in the receptor binding domain might actually give that virus an advantage and allow it to infect some other host. So that's a great question. The role of selection and really which viruses make it and which ones don't after they've recombined is a really good question. Another thing that I think that you're kind of bringing up here is the direction of recombination, and correct me if I'm wrong, but when you think of recombination from a phylogenetic standpoint, it could go either way. So if you just have the polymerase tree and you just have the receptor binding domain tree, there's actually no way to tell if the polymerase was the one that recombined or if the receptor binding domain was the one that recombined. So in our case, we think that there actually is evidence for the direction, and that would be given that the ACE2 using clade is the ancestral clade. And so it just doesn't make sense that the recombination could have gone in the opposite direction. And then, we just talked about this yesterday, Simon, but I forgot the second part.
If I think of it, I'll let you know.
Yeah, it's in my notes somewhere.
No, it's a really important distinction, though. And so in just the mutational mechanisms, because we're always somehow drawn to the idea, I think, of point mutations as being the real sort of energy of the evolutionary process. And yet, and there's evidence of that, right? So, Vincent, we've talked at some length about a single point mutation at position 614, I think, of the spike protein and a single amino acid substitution that sort of swept through most of the pandemic so far. And if and whether there's reason to be alarmed by that, it's really kind of caught the attention. Whereas there's certainly a lot of people are looking at recombination, but it doesn't make the news headlines kind of in the same way. It's really interesting. Whereas I think when you really think about the virology here, that it's incredible the amount of recombination happening, and yet that's sort of it's harder to get a handle on, just as we're discussing this, right? And so I think it gets a little bit relegated to the background. So really important to consider these ideas.
It's definitely a scale problem. So, single point mutations have effects that make sense to us. They'll make binding a bit better, or they might make binding a bit worse. Whereas recombination is kind of, it's on a different scale. It encompasses multiple point mutations. There might be a bunch of different points in the genome that are different between the two viruses that are recombining. So understanding the impacts of those collection of differences versus like one point mutation is a different scale. And it's something that's a lot harder to think about and to measure and to predict, what's going to happen based on that. So yeah, I think that you're right. It's sometimes tempting or easier to focus on single point mutations. And those do matter. Like Simon was saying in the Ebola example, and that's Kartik's eLife paper actually is where this was first shown, was that a single amino acid change in the NPC1 receptor was able to pretty much abolish Ebola binding in the straw-colored fruit bat. And so there's a scale of affinity that might be affected by point mutations, but then there's also like an even wider scale of possibility. So is it even possible to bind ACE2? And then a smaller scale of like, how well do you bind ACE2? Right. So there's different levels here that we're kind of discussing things on when we talk about recombination versus point mutations, or when we talk about ACE2 usage in general versus human ACE2 usage. So disentangling those two scales is also really important and necessary for framing discussions like this.
Just a side note on recombination. This recombination, of course, requires that a single cell be multiply infected. Is there evidence that bats have many different Sarbecoviruses in them? Does the sampling support that?
Well, we find we do find in the global surveillance efforts that we've been involved in. It's not very often that we detect co-infections, but we do find them. What we can't know with the sort of sampling that we do is whether or not a single cell was co-infected, or if the animal just happened to be.
It would have to be a cell, because otherwise you don't get the recombination. But it has to start in the animal. So I'm guessing that in these caves, there are many, many viruses in each bat, and they're always swapping them, and that gives you the diversity for sure.
Yeah. So that's kind of part of the reason why the phylogeography of this study was so important. So that figure 1, where we showed that there's a geographic breakdown between these different groups of viruses, once you get to SARS-CoV-2 and that group, they actually overlap with a group of viruses that already exists. So they break down that pattern where each group has its own geographic area, and they actually kind of just swap right into Yunnan province and the surrounding region, which is the same region where SARS-1 and its relatives were found. So this is essentially ecological evidence, right? That there is potential for these viruses to be circulating in the same geographic area, and that's necessary for them to be infecting the same host individual at the same time and to recombine. So we use that phylogeographic evidence as support for this recombination hypothesis, because without that overlap in geographic space, you can't have recombination.
Sure. Is there any role for non-bat species, non-bat mammal species. So we learned this because of this pandemic that pangolins can be infected, and they have the same ACE2 binding RBD, right? So are they just dead ends from bats, or are other animals involved? Do we have any idea? Is anyone looking?
I'm sure there are more species that are going to have these Sarbecoviruses, and probably now, like the pangolins, have their own discrete lineages that sort of have stemmed from a prior spillover event. We know there's some ecological evidence now that coming out where showing that bats and pangolins are sort of cohabiting in these sort of tree cavities, and of course there are other species that probably cohabit too. And so, looking at the ecology of those discrete systems, I think would be very informative as to thinking how often this sort of thing actually occurs.
I think it's so important to I'm sorry, Nels. It's so important to make sure the virologists understand how important the ecology is, right? They can't work in vacuums.
Right.
Well, so Simon, you're mentioning these sort of potentially natural collisions in kind of cohabitating pangolins and bats. But then, there's also, we hear a lot about the increase in the wildlife trade, some of it illicit. And I think you even have a phrase in here, the anthropogenically transported scenarios. So can you give us a sense of the scale of this? We hear about it all the time, that, in some of the markets, Southeast Asia, et cetera. Does that rival the kind of natural collisions between different species, or what's the scale of that?
It's a difficult question to answer. Obviously, the wildlife trade is, I think I'm correct in saying, like second to the, so there's the legal trade in wildlife, of course, and then there's also the illegal trade in wildlife. And I think that, it rivals drugs in terms of the illegal, the illegal trade. And so it's really, really a significant problem. And not just for our health, it's for the conservation of these species foremost. So the scale of the problem is massive. And because many of these animals are live and they're mixing all along the way, we don't know the viruses that they have. There's always the potential for viruses to spill over into intermediate hosts and end up in people or directly into people. I don't have a good answer for you, other than to say it's a big problem. And needs to be studied.
Nels, when we had Peter Daszak on TWiEVO a couple of weeks ago, he says it's a massive problem. You could, in parts of China, you can just dial on your cell phone what kind of wildlife you want to eat, and they bring it to you in the restaurant, right? A few days later. I
heard that episode, and big recommend for our listeners, if you haven't already, to cross over to the TWiEVO, and that one in particular, I think, hearing Peter tell some of these stories, it also circles back to some of the issues we've been raising about funding and sampling, because the coronaviruses are still massively undersampled, and some really scary political shadows on how we fund science, how that can get wrapped up. I think it's a really important conversation, and I would definitely encourage, if you haven't already, seeking out that TWiEVO episode. Among the sea of TWiEVO episodes. Do you guys, Vincent, is it like 5 times a week now that you do it?
Two times a week now.
That's great. Yeah.
But
Pete, so maybe
go ahead.
Go ahead.
Simon, are coronas the only viruses we need to worry about in wildlife?
No, there's certainly, of course, something that we need to worry about right now as we're dealing with this. But as we've learned, even over the last 20 years, it'll be something else the next time round. And it's sort of a difficult message to sort of give when you're in the middle of a pandemic, to remind people that this is not the only coronavirus, or even not the only wildlife virus that has the potential to do this or worse. So it's important that we continue to do this type of work. It's important that we, not just the viral discovery and the viral characterization, but community engagement and ecology and working with policymakers, the whole gamut of things that ultimately are required to sort of build better pandemic prevention.
Yeah, and I pulled out two papers of yours. One, you've got herpes viruses from bats in Puerto Rican caves, and then another one where you have adenoviruses and herpes viruses from great apes, just as examples, right?
Right. Yeah, and so they're interesting because they're sort of studies that are, at least the sort of the Puerto Rican caves is a study by Anna Shoden, and that's interesting because there she's really starting to pull in theories and concepts from community ecology and trying to understand, at a very fundamental level, like what are those mechanisms that sort of lead to viral communities being assembled in these different hosts within a cave system where you have multiple species co-roosting. Is it about the sort of the genetics or the traits of those individual hosts that are important? Or is it something else? Is it just about the opportunity that they're all mixed in there together? What are those genetic and ecological processes that ultimately allow viruses to exist in the hosts that they do?
Nels, anything else from you?
Sorry. Yes. So I think we're going to probably wrap up pretty soon. We could talk for hours as usual here, as we're just starting to dig in. We've skimped a little bit on sort of the last part of the manuscript, which is going from it, just as Heather is describing. So, the way that a single recombination event, unlike a point mutation, could bring in sort of a lot of diversity to the rest of the virus genome sort of in one go. And so, that in the manuscript goes there, right? So we see in the receptor binding domain in particular, how there's this diversity that's built up through these recombination events in the comparison between the different viruses that were recovered from the bats from Africa versus the ones in Southeast Asia. And then even going one more step. So we're all the way from, collecting these viruses from mysterious bat species in Africa to actually testing whether the receptor binding domain can recognize in a VSV pseudotyped experiment, recognize the human ACE2 receptor. So maybe we could wrap up just with that, maybe a quick summary of that part of the study, Heather, and how that how that happened. How do you do that all the way from the field to the sort of petri dish?
Yeah, that's a really good question. And I think I'll maybe give an introduction and give a stab at this and then pass it over to Simon, because he's more of our virologist lab expert and I'm more of the math and ecology girl. So but yeah, this is a really important system because you would like to be able to study viruses just as they are, as a whole virus, and test its ability to infect humans. But you can't experiment on humans, first of all. And having these live viruses in the lab is not typically super safe. So the way that we kind of get around that and are able to study these viruses in a very reductionistic way, but in a safe way in a BSL-2 laboratory, is by taking that spike protein from the coronavirus. And as I mentioned before, that spike protein contains the receptor binding domain, which is the determinant of whether or not the virus can take that first step of binding a host cell. So you can take that spike protein out and stick it into another virus that is pathogenically safe and not dangerous to people, which is VSV, vesicular stomatitis virus, which is a mouse virus. So you can take the spike out of the coronavirus, put it into the VSV virus, then you don't have any of the dangerous parts of the coronavirus, like the things that are going to cause symptoms and disease and evade the immune system and things like that. And now you have the ability to test that spike protein's affinity for binding human cells without any of the potential dangerous implications of whole virus infection. So what they've done, what Michael Letko has done is basically that. Take this spike backbone. What he did is he took the spike backbone of SARS-1, and then he specifically swapped out just the receptor binding domain section. So just the section that actually makes contact with ACE2. So the SARS-1 spike backbone inside a VSV virus with the receptor binding domain from the strains that we found in Africa. So he took these, I don't even know what you call them, like Frankenstein viruses, and
Not Frankenstein viruses. Let's use very non-scary terminology.
Fair enough. You're right. That's a scary
surrogate
word.
pseudotyping system.
Yes. After I just explained how not dangerous they are, I call them Frankenstein viruses. But yeah, we take these. What did you call them? Pseudotyped. Surrogate pseudotype. This is not my area of expertise.
You're doing very well. Pseudotyped. For a math ecology girl, you're doing very well.
So surrogate pseudotyped systems allow us to study this binding interaction between the virus and human cells in a safe and controlled way. So basically, they took this VSV virus with the receptor binding domain from the viruses from Africa and tested it in a BHK cell line, which was expressing human ACE2. So BHK is hamster kidney cells, I think. Mm-hmm. So importantly, there was no binding in the negative control. So these hamster kidney cells don't express any kind of ACE2 that is able to bind to SARS or to the African strains. So just to get that out of the way, there's no binding to the hamster cell itself. And only when you specifically force those cells to express human ACE2 do you see binding. So then on the positive control, you can see that there's binding when you express human ACE2 in these BHK cells between the VSV SARS-1 virus. However, when you swap out that receptor binding domain and then you test it with the African strains from Uganda and Rwanda, you see that there's no binding again. So that leads to the conclusion, logically, that the Rwanda and Uganda viruses are not able to bind human ACE2.
But they must bind something for you to be able to grow up the VSV pseudotype, right? Because you've substituted the coronavirus spike for the VSV one, I presume. So what kind of cells are the viruses grown in, do you know? Human cells?
They are... Where is that? I can tell you that. They are grown in 293 T cells.
So presumably some non-ACE2 protein is acting as a receptor for those, yeah, which you could identify if you were interested in.
Yeah.
Would be interesting.
So I guess we can maybe rest a little bit more comfortably that at least these two strains or this family that you've sampled in this diversity are not going to recognize human ACE2 in the same way that SARS does. And so,
yeah.
But as
and evolutionarily, so did they lose that ability, or did they sort of gain, or how do you think about that one? We kind of talked about that a little bit at the top.
Yeah,
so path one way or another here.
Yeah, and Simon kind of alluded to this, and we kind of talked about it when we discussed like scales and gradients of affinity. But it's a really important question that we don't actually know the answer to yet. Whether that ACE2 binding is abolished because of specific point mutations, and then presumably it could potentially come back if those point mutations are swapped, or is it because of the structure of that receptor binding domain in the viruses from Africa that they have different amino acid point mutations, but they also have a small deletion in what we are calling region 2, just for clarity. But is it that deletion that abolishes binding in these viruses? Is it the differences in the receptor binding ridge, which is also in one of our figures in the paper? So we don't actually know which of these differences is responsible for the lack of binding. And it would be really nice. It would make a really nice story if it was the deletion, because then we could say, okay, we have these stepwise deletions where first there was sequence that was lost in region 2, and then viruses diversified into Africa and Europe. And then subsequently, sequence was lost in the receptor binding domain, and then viruses diversified across China. It makes a really nice story. We don't necessarily know that that's the case. But if it is the deletion that abrogates binding, then, that's really good evidence for the stepwise hypothesis. And, I think makes a really nice evolutionary story. But yeah, we don't know that for sure. We don't know whether they're still using that ACE2 or whether that deletion has pushed them into using some other receptor. Whereas with the viruses in Asia, in China specifically, that have the larger deletion in the receptor binding domain, we can be quite confident that those are not using ACE2. Mm-hmm. But with the African ones, it's a slightly different story. It's still kind of muddled. There's a lot to be figured out yet.
Yeah, and I have one last question. I just can't resist this one. So the last evidence sort of presented in the final figure is some analysis of positive selection. And kind of intriguingly divided it into two groups. So the spikes from viruses that use ACE2, and then the ones from viruses that don't use ACE2 and have those deletions that you were just discussing. So here it looks like there's some signal, some evidence perhaps, that the ones that recognize ACE2 show greater diversification or show positive selection, sort of signals of recurrent changes happening more so than the ones that don't. And so it's kind of interesting because I think there is some good evidence that on the host side, ACE2, at least among the bats, that there is positive selection sort of that would get to this potential idea of an arms race. So I'm curious, maybe, and we just kind of hinted at that, but maybe to just go into that a little bit. And I'm curious about your thoughts about how these differences in the evolution between the two groups.
Yeah, this is another really important thing that we started investigating for one reason and then ended up making different conclusions based on what we found, or generating new hypotheses based on what we found. So the initial reason that I started looking into positive selection between these two groups was because I wanted to, not that I wanted to disprove, because I know you're not supposed to have pet hypotheses, but I was interested in evidence that would potentially suggest that convergent evolution was not the case here. And I was thinking, well, what kind of signatures would you see in the genome if there was convergent evolution towards a particular phenotype? And selection was one of the things that came to mind. So I decided to look for selection and to look in these two different groups and to see if there were selective pressures in the same regions of the genome, because that could potentially explain how a non-ACE2 user might be experiencing selection to become an ACE2 user. But we didn't find that. In the non-ACE2 users in the receptor binding domain, where we expect to see positive selection due to this evolutionary arms race with the immune system, we didn't see evidence of positive selection in the non-ACE2 users, which to us was evidence that there's probably not a lot of selective pressure for convergence to the ACE2 using trait. But secondarily to that, it signaled to me, and this is kind of like my pet idea, and it's again, it's another hypothesis, another idea that we've come up with from this evidence. But typically when you have a receptor binding domain, positive selection in that domain is common, is almost always present because of this arms race. And because that positive selection is almost completely absent in the receptor binding domain in the non-ACE2 users, at least in the receptor binding domain for the ACE2 users, that suggested to me potentially that that might not be the location of the receptor binding domain. So coronaviruses are known to have receptor binding domains in either the C-terminal site or the N-terminal site of S1. It can be in a different part of the protein. And so I kind of was thinking that this positive selection evidence is also supportive of the idea that these non-ACE2 users are probably not using ACE2 at all. It's not that they're just using a different bat ACE2, it's they're not using ACE2 at all. And that receptor binding domain is potentially in a completely different part of the spike protein.
Yeah, it would be cool to take the RBD out of those pseudotyped viruses and see if they still grow in 293 cells, which would, if they did, that means that's not the binding part anyway. It's a long shot, but...
Also be super exciting, I think, to start getting more ACE2 diversity on the bat side in particular, and see if some of the signals of diversifying selection could actually sort of have functional outcomes. That's my pet hypothesis, if I have to confess one, which is that the ACE2 users have been sort of ramped up on genetic diversity, and then the chance that you land, kind of as we discussed, on the human, just sort of as a sort of side effect, kind of increases somehow. And that there's even an old African proverb that gets at this idea. And it maybe even ties back to Simon's original occupation that he was considering. And that's when two elephants fight, it's the grass that suffers. And in this analogy, grass is us, the humans who are enduring a global pandemic and have sort of retooled all our conversations into this direction. So anyway, I'll rest my case there with that.
That's a good way to stop, Nels. I think we should stop. We've kept these two long enough.
Yeah, really fun.
This has been TWiEVO 58. You can find the show notes at microbe.tv/TWiEVO. If you want to send in a question or comment, TWiEVO at microbe.tv. If you like what we do, consider supporting us. Microbe.tv/contribute. Nell Zelzi is at cellvolution.org on Twitter. Earlybird. Thanks, Nels.
Yeah, thank you, Vincent. Great to be together. We kind of had an impromptu summer break, including a week delay. So this is actually our July episode coming out in mid-August, but I think we'll try to turn one around quickly and get back into it. Really fun to be back and talking science with you, Vincent, and our guests today. Thank you all.
Our guest from Center for Infection and Immunity at School of Public Health at Columbia, Simon Anthony. Thanks for joining us, Simon.
Thank you.
And Heather Wells, thank you.
Thank you, Vincent. Thank you for having me.
So you have a couple of years left in your PhD, right?
I wouldn't say a couple. I would say one.
One, okay. What are you going to do next?
I don't know, but I'm having such a good time thinking about these problems and really churning through these ideas. And I can't see giving up on coronaviruses. So definitely probably just continuing down the path. But we'll see.
All right.
That's great. Oh, and I can't help but think, Vincent, just from our discussion, I'm hoping there's a lot more rising stars like Heather out there in the Midwest who are trying to convince their parents that rodents or llamas or whatever are really cool and that you should study them because there will be more viruses. There will be more spillovers, and it won't just be coronaviruses.
Yeah, I mean, I was thinking during our conversation, we need to be very convincing because we don't control the money and we have to convince the people that do that these things we're talking about today have to be supported on a big scale. And it takes eloquent people to put it in words that convince people. It's not easy to do, right? But that's what we have to do. I'm Vincent Racaniello. You can find me at virology.blog. Music on TWiEVO is by Trampled by Turtles. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Till then, stay curious.